So good morning to this HEAS Seminar for Genomics. Today I'm happy to present Irina Velsko. Irina got her PhD at the University of Florida College of Medicine. Her project looked at the association between periodontal disease and atherosclerosis, specifically how the bacteria responsible for gum disease can be directly involved in atherosclerosis development. She has done postdoc at the University of Florida, investigating how oral biofilm interacts with blood cells to induce immune responses at the University of Oxford, working on proteomics and metagenomics of ancient dental calculus, at Clemson University studying the genomics of oral Streptococcus species, and is now at the Max Planck Institute for Evolutionary Anthropology, where she has multiple projects investigating oral microbiome evolution using ancient dental calculus. And today she will be talking about expanding known diversity of human-associated microbes through assembly of ancient dental calculus. I'm very happy to have you.
Thank you for inviting me. All right. So as Miriam said, I'm Irina Velsko, and I'll be talking to you today about expanding known diversity of human-associated microbes through assembly of ancient dental calculus. So the oral microbiome is estimated to consist of approximately 700 microbial species, mostly bacterial and archaeal. And historically, these species have been studied through laboratory culture-based techniques, but this has limited us to being able to study only those organisms which we can actually grow up in the lab. Many of the oral microbes are fastidious organisms. This means they have very specific and particular growth requirements, and so the media that we need to make to grow them up can be very complex. It can involve very unusual reagents, and it can be very expensive. So studying these organisms tends to be limited to labs that are very specifically focused on oral microbes. In addition to this, many of them grow very, very slowly, and that makes them difficult to study with the standard microbiology techniques that we use. Further, many of these species also have unknown metabolic requirements, and so we just can't make up media to actually get them to grow in the lab, and so we're unable to grow them at all and study them through this method. So for these reasons, many oral microbes actually remain difficult to impossible to culture and study within the lab. So the introduction of metagenomics really allowed us to expand the known diversity of oral microbes because we're able to identify the species that are present based on their genomic content, and we don't have to be able to grow them up to be able to know that they're there. So the introduction of metagenomics is really helpful for us to expand the known diversity of oral microbes. And early metagenomic studies are where we get this estimation of approximately 700 different bacterial and archaeal species within the oral microbiome. But metagenomics itself does have some limitations, and that is that we can only identify species which are in a metagenomic sample if we have a reference genome currently in our database. And this basically limits us to the organisms that we can culture up and grow to extract their DNA. So when we do taxonomic profiling of a metagenome sample, we'll take the short reads of our metagenome and we'll match them up to the genomes of the species which are in our databases. When we do that, some of the reads will match up to those genomes, some of them will not. And at the end, we can count up all of the reads that match to each of the species within our databases, and we can put them into a table where we either have the total number of reads per species per sample or a relative abundance of each species per sample. And then we move forward with this abundance table to do our diversity analysis calculations to see what are differences between different groups that we're interested in. But there's always a group of reads that are not taxonomically classified. And this means that we don't know what they're coming from. We don't know what species they're representing. And so they're data that we can't use. And we've just historically done nothing with them. We ignore them because if we don't know what they're coming from, we can't do anything with them. But how much of a problem is this for us, particularly within ancient dental calculus? So this is something that I looked into across a variety of oral samples. So I'm showing here the percentage of reads that were classified in a variety of oral samples, which I've grouped across the x-axis by the study that they come from. And then across the top, they're grouped by the oral source. So I have a bunch of studies from ancient dental calculus, as well as a bunch of modern oral samples, including calculus from living populations in Spain, plaque, buccal mucosa, which are cheek swabs, and saliva samples from both industrial and non-industrial populations. And what we can see with the ancient calculus samples is that on average, these samples have fewer than 50% of their reads taxonomically classified, which is a really small amount of the data to be using for generating our species tables. In contrast, most of the modern data that you see here has about 75% of the reads taxonomically classified, so we're able to use much more of the data in these samples and hopefully get a much more accurate species profile. So how much of a problem is it within ancient dental calculus that we're using so little of our data? We're only able to classify generally less than 50% of it. So does this affect, or how does this affect the species profiles that we're getting? So this is something that I looked into with a project where I was looking at ancient dental calculus from the Pacific, and I included samples from a variety of other studies as well. So within this project, I was trying to see whether or not the species community within ancient calculus samples from the Pacific were distinct in any way from the species communities and samples from across the globe. So I had samples from Africa, Asia, the Caribbean, North America as my comparative samples here. And I looked at the diversity differences through a beta diversity analysis in a PCA, which I plotted here, where each point represents a calculus sample. And if the points plot closer together, it means that the species community is more similar. If they plot more distantly, it means that the species community is more distinct. What I was hoping to see was that the samples would cluster together based on their geographic origin, but that's not what we see here. It's generally a sort of indistinct blob, which is actually fairly typical of what we see with calculus when we perform this analysis. And no matter where the studies come from or where the samples come from. But it does look like many of the samples from the Pacific do cluster at one end of this plot in very negative PC1 values along the x-axis. So there may in fact be some kind of diversity difference within these samples compared to the other samples within the study group here. So I wanted to look into what this might be. And my first thought was, well, maybe there's a difference in the number of species that are found in these samples compared to the others. So I looked into the number of species that were found in each of these samples. And I plotted that here, where I have the samples again plotted along the x-axis with their PC1 loading. So the x-axis is identical between the upper and lower plots. But on the y-axis here, I have the number of species that are identified in each of these samples. And you can see there's clearly a trend where the samples that have very negative PC1 loadings also have fewer species identified than the samples that have more positive PC1 loadings. So it looks like what we're seeing here is that these samples in very far negative PC1 loadings have lower diversity. They have fewer species than samples that plot in more positive values. But I wanted to confirm that this was actually the case and that we weren't missing some data here. So I also looked at the percentage of reads that were taxonomically classified in each of these samples. And that I'm showing here now with the color of these points. So the reads that have a higher taxonomic classification are a brighter yellow color, and the reads with lower taxonomic classification rate are a darker purple color. And you can see there's also a trend where the samples with fewer species have fewer reads which are taxonomically classified than the samples that have more reads that are taxonomically classified. So what it looks like we're seeing here is actually not a difference in the diversity of our samples, but an inability to identify the diversity in our samples. So if we classify fewer reads within a group of samples, then we don't actually know how many species are genuinely present. We very likely have one or more that are just not in our database, and so we can't identify them. And so we get a lower taxonomic assignment rate, we get fewer species returned at the end of our classification process. So while we might be tempted to say initially that these samples are less diverse, what we can actually say is we don't know how diverse they are because we don't know what we're missing. So we can't actually draw any nice conclusions about the diversity differences within these sample types, unfortunately. And this sorts of issue leaves us hanging, and we can't really form the conclusions about the studies that we would actually like to draw because we just don't have sufficient data to do so. So it's important to note when we work with data like this that technical limitations, in this case, an inability to identify all of the diversity in our samples, can appear as a biological trend. So it makes it look like our samples are less diverse, but this is not actually the case. And so when we have this problem, we can't actually appropriately interpret biological diversity differences between sample groups because we don't have an appropriate measurement of diversity. So within calculus studies, ancient calculus studies in particular, we want to try to understand how we can get around this problem. How can we get better diversity estimates? And the first way that we approach this is to try to understand why are we getting low taxonomic assignment rates within our ancient calculus samples. So there's a couple of possibilities. The first one is that it's an artifact of working with ancient DNA. These reads are quite short and they have specific damage patterns, and both of these could possibly interfere with our ability to taxonomically classify the reads. We actually know from several studies that the damage patterns on DNA do not impact taxonomic classification, so that's a good thing for us. But read length does impact it. Shorter reads cannot be classified because there's just not enough information in them. They match up to too many genomes because they're non-specific, and so taxonomic profilers will leave them unclassified because they don't know which genome they actually originally came from. So it's certainly a problem that we have with ancient DNA. Our reads are much shorter than we have with modern DNA, and we will have some reads that just can't be classified because they are too short. But there are plenty of reads in these samples that have no taxonomic classification, and yet they are just as long as reads that do have taxonomic classification. So clearly read length is not the only problem that we have with our taxonomic assignment rates in ancient calculus. So another possibility is that we have a sampling bias in constructing our reference databases. And this means that there are species within our ancient calculus samples which exist in living populations across the globe today, but we have not yet sampled those populations. We have not yet sequenced those microbiomes. And so we don't have representative genomes of those species yet present in our databases. But with broader global sampling efforts, we will be able to then sequence up those organisms, put them in our databases, and we should be able to improve our taxonomic classification rate. Another, well, we actually do have evidence that this is probably very likely the case, because with gut studies, we've seen that as more broad global populations are sampled, we do see increasing numbers of species which are present in these individuals that are not currently in our reference databases. And so this is very likely going to be the case with oral samples as well, although they have not been extensively studied in the same way. And another possibility is that some of the species present within our ancient calculus samples are actually extinct species, and they no longer exist in any living population anywhere across the globe today. No matter how much sampling and sequencing we do, we'll never recover them because they just don't exist anymore. And this is something that is quite intriguing for us, but it's much more difficult to address. But all three of these can be basically wrapped up into the single question of how can we know what we're missing in our data? And the field of metagenomics and microbiome studies has turned to de novo assembly of metagenomes to try to answer this by directly reconstructing genomes from a metagenome sample rather than relying on references to do so. So in de novo assembly of metagenomes, essentially what we do is we take the short reads in our metagenome sample and we align them to themselves, and we hope that all of the short reads from the same original genomes will align with each other. And then we call a consensus sequence on these alignments to generate very long sequences called contigs. And then we have another program that takes those contigs and it will group them together, hopefully getting all of the contigs from the same original genomes into distinct bins. And these bins hopefully represent the full array of the genomes which were originally present within our metagenome sample. Now, through this process, we call the final stage a bin, but when we start to work with them as genomes, we call them mags for metagenome assembled genomes. And once we have our set of mags from our metagenomes, we can try to identify whether or not we have known or unknown species within these mags by comparing them then to our reference databases and seeing how well they match up to what we currently have. So when we do taxonomic identification of our mags, we generally get three kinds of taxonomic identification returned. And the first kind is ones for which we have species that are present already in NCBI. And these, we can usually get a full genus and species name of these mags. So we would get something like Tannerella forsythia, Actinomyces auris, Prevotella denticola. Basically, these mags represent novel strains of these species, but not novel species themselves. The second kind of classification that we get are ones for which there's really only a close relative in NCBI. And in cases like this, the taxonomic profiler can identify that this mag belongs within a particular genus, but that mag is different enough from all of the other species which are in that genus that it can't give it its taxonomic name. And so it leaves the species blank, but it will tell us that it belongs within that genus. So we'll get a returned taxonomic identification that's something like Microbium species blank. And the third kind of taxonomic classification that we tend to get are those for which there's only a distant relative present in NCBI. And in this case, the taxonomic profiler can identify that this mag belongs within a higher taxonomic level, such as a family or a class, but that mag is different enough from all of the other species within that family or class that it cannot assign a species name. So we'll get something like this, where we have a family name Vianelaceae, but the genus is blank and the species is blank. Now, of these taxonomic classifications, the ones that are of the most interest to us are the ones that don't have a species name, because these likely represent novel taxa in our samples. And this is what we're really interested in trying to understand how much diversity exists within our ancient samples. But we do have a problem with this kind of classification, and that is that we don't know how many species are actually represented when we can't assign it the species name through this method. So for example, if we have multiple mags with the same species blank name, so Microbium species blank, we don't know if all of those mags represent the same novel species or whether there are actually multiple species within this group of mags. And so we need a way to disentangle that to be able to truly understand how much novel diversity exists within our samples. So how do we begin to start assigning species names and disentangling diversity when we have novel mags potentially representing novel species that aren't assigned a species name through this method? So I'll talk briefly about a case study in which I've been doing this using an organism called Anaerolinaceae bacterium oral taxon 439, or Abbit 439 for short. So Anaerolinaceae bacterium oral taxon 439 is an organism that's conserved across the dental calculus across the primate lineage. So it can be found in humans, chimpanzees, gorillas, and howler monkeys. So it's one that's been evolving along with the human lineage for a very long time. It's unfortunately never been given a formal genus and species name. So when the group that sequenced the genome for this organism deposited it in NCBI, they left it essentially with a placeholder name. So they matched it to the genus or to the family Anaerolinaceae, but they didn't give it a genus and species name. So they just gave it this essential placeholder name of Bacterium oral taxon 439, which makes it a little bit difficult to work with, but that's what we have for now. So this organism is quite abundant within ancient dental calculus, but it's quite rare to detect within modern industrialized plaque samples. So those from populations living in Europe and North America. And it's one of these that's extremely difficult for us to culture in the lab. We actually don't know what its growth requirements are. And so to get it to grow in the lab, you actually have to grow it with a second species on the same plate, because that second species produces something, but we don't know what, that will allow this one to grow on a plate as well. But that means that we can't easily study it because we have two species growing on the same plate, and we can't really disentangle who's doing what for our microbiological studies in that case. So because it's rare and because it's difficult to culture, we know very little about its biochemistry and physiology. It has not been extensively studied. It's very little that has been done to try to study this organism. And so there's only one isolate genome from this organism that's available within NCBI. So we also know very little about the genetic and genomic diversity of this particular organism. However, because it's so prevalent and so abundant within ancient dental calculus, it's become extremely popular for studies to map their ancient dental calculus samples against this one reference genome to try to reconstruct phylogenies of this organism to look at its evolutionary history. Many of the studies that do this, however, have reported that there are high levels of heterozygous SNPs in their mapping for many of their calculus samples, which suggests that there's actually multiple closely related species or multiple strains of this organism that are present in these calculus samples. But if there's only one reference genome available, how can we understand how much diversity actually exists within individual calculus samples and across our calculus samples? And so this is the kind of question that we can address by using de novo assembly from these calculus samples to produce MAGs to try to get an actual idea of the true diversity within our samples. So this is something that I've been working on. I have a set of ancient and modern dental calculus MAGs that were produced by my colleague, and then I assembled a selection of modern dental plaque samples which come from populations who live in Cameroon, who have very low integration into the global market economy and would be called non-industrial populations in microbiome, I guess, colloquial speak. So sifting through all of the MAGs that were produced from assemblies of all of these samples, I was able to identify 300 that were very likely Abbit 439 or some organism that was closely related to it. 276 of these MAGs came from our ancient calculus samples, 9 from modern calculus samples, and 15 came from these modern plaque samples from Cameroon. Now, the naming of bacteria can be messy, and as I mentioned, this one is rather messy. It's particularly messy. So within NCBI, this genome is designated as Abbit 439, but the program that we use for taxonomic classification is actually the GTDB, for the Genome Taxonomy Database. And what this program did was they went through NCBI and they tried to clarify taxonomy across all of the bacterial species there based on genome similarity to make it much more cleaner and easier to work with. And so GTDB has reclassified Abbit 439 into the genus Flexolinea, and GTDB does not create a species name if there isn't one in NCBI. So instead what they'll do is they'll take the genome accession from NCBI and they'll use that as a species placeholder. So within GTDB, when we see Flexolinea species 0017175545, this refers to the Abbit 439 reference genome directly. So within this set of 300 MAGs, I had 218 MAGs that had this species-specific designation, but I also had 82 MAGs which had no species designation. So there's at least 2 distinct species present within these MAGs, which is already more than the currently known 1 species. But how many species are actually present there? Is it possible that there are more species represented by these unnamed Flexolinea species blank than just 2? So to answer this question, I clustered the MAGs based on the average nucleotide identity using a cutoff of 95% to define species boundaries, because this is a standard that's used within microbiology. And this clustering analysis revealed that there are in fact 10 distinct species clusters within this 300 MAGs. So we've massively expanded the known diversity of this organism from 1 species now to 10 distinct species. So there's actually a lot of previously unrecognized diversity within this group of organisms within the mouth. 8 of those species clusters consisted only of MAGs that had no species designation. 1 cluster consisted entirely of MAGs with the reference species designation, and then 1 cluster was a mix of MAGs with and without the species designation. So we did have a lot of unrecognized diversity within each of these species clusters. But this is a rather surprising result to see so much unrealized diversity within these samples. So I wanted to confirm this, so I generated a core genome phylogeny of these MAGs, which I've shown here. And I've colored the tips of this phylogeny by the species cluster that each of these mags fell into based on the genome nucleotide identity clustering. And it turns out that those species cluster fall really neatly onto the branching patterns of this core genome phylogeny. So it really well supports that we do in fact have 9 novel undescribed species within this group of organisms now, which we had no idea existed. And had we never gone with metagenome assembly to do this, we would never have known that these actually exist within these calculus samples. So we now have 9 novel species, plus many representatives of strains of each of these species that we can work with our taxonomic profiling. We can hopefully get better diversity estimates, particularly regarding this group of organisms within our calculus samples. We can hopefully map our calculus samples to a more appropriate reference genome so we can build more accurate phylogenies and look at the study of these organisms, either one in particular or this group of organisms within our calculus samples. So this goes to show that there really is a substantial amount of unrecognized diversity currently within oral microbiome samples, both ancient and modern, which had not been not recognized before. But is this just a one-off example? Did we just happen to land on one that has a whole lot of unknown diversity and this was not actually common across oral taxa? Or does it turn out that there's actually quite a lot of unrecognized diversity within the oral microbiome? And so this is a question that I've been looking at much more broadly than with just ancient samples, but across geography and across temporal sample selection, how much undescribed diversity does exist in oral microbiome samples? So how many species do we see within MAGs that are not represented by reference genomes present today? So to answer this question, I really did want to take a very broad approach, and I wanted to use as many sample types as I could from the mouth and from as much of a geographic representation as I could get. So I collected together all of the MAGs that I could that came from ancient and modern dental calculus, plaque samples, buccal mucosa samples, saliva samples, and tongue samples from living populations across the globe that are both industrial and non-industrial. So that there is a decent global representation from these samples, although we could always do with more of each type from across the globe to get better sample representation. And these samples do come from numerous individuals, so we don't have just a single individual sampled, which is important for getting a good representation of diversity within and across populations, both globally and temporally. So altogether, the MAGs that I was able to collect from these samples from these individuals totaled up to about 24,000 genomes, which is a very big number. It's a lot to work with. And so I needed some way to reduce this number down to something that's a little bit more easy to work with. So I decided the first step would be to see just how many species are actually represented by these 24,000 MAGs. So I clustered these genomes in the same way that I clustered the Abbit 439 genomes based on the genome identity with a 95% cutoff to define species boundaries. And when I did this clustering, I ended up with nearly 4,000 distinct species, which is a really high number given that we had estimated there were approximately 700 species that live in the mouth. So this is much, much higher than that. But there are some ancient calculus samples in this data set that come from non-humans. So perhaps part of that inflation comes from looking at samples that aren't human. So if I pull out only the species that were found within human samples, well, the number doesn't actually go down that much. We now have about 3,400 species which are present in human samples, which is still a very big increase from the estimated 700 known species that we started out this project knowing about. And it turns out of these 3,400 species, 2,500 have no species names, which is a huge amount of previously unrecognized diversity in the oral cavity. So is this number reasonable based on the data that we have? Well, I did have to remove 217 MAGs, which are likely from the environment. So it's impossible to completely clean off ancient dental calculus before the extraction. And so it's very likely that these came from dirt samples, which were still part of the calculus when we did the extraction. It's not surprising. It's very clear that they do come from the environment. They're very distinct from organisms that we find within humans, and so they were pretty easy to remove. And this left us with 664 named species within our collection, which is quite close to the estimated 700 known species within the mouth. So it does seem fairly reasonable that we do have now 2,500 new species, which we've only been able to identify because we have MAGs, and we have MAGs both temporally and globally from diverse data sets. So this is a really big increase in the known diversity. So how can this help us with performing diversity analyses, particularly regarding our ancient calculus samples? So does adding this novel diversity to our taxonomic databases improve our taxonomic assignment rates? So this is a regraphing of the data that I showed earlier in the presentation, where I have the average or the percentage of reads that were classified per sample, just of my ancient calculus samples, this time with the studies grouped across the bottom, and I've added several more of the ancient sample studies to this plot. But the database that I was using is the Genome Taxonomy database, which is basically the most comprehensive bacterial and archaeal database that exists today. It includes all of the bacterial and archaeal genomes that are present in NCBI. It has about 63,000 genomes, and it has basically all of the known characterized microbial diversity. And when we use this database, we have generally less than 50% of our reads taxonomically classified. So even though this is the most comprehensive database we have, we're missing out on a lot of diversity in these samples. Now, within oral microbiome research, we're very fortunate to have what's called the Human Oral Microbiome Database, which is a collection of all of the genomes that represent taxa that are known to exist in the mouth, which was built off of this original metagenome study that did estimate there were approximately 700 species in the mouth. So I thought, what I want to see is if we profile our samples with just the known oral diversity, what percentage of reads are classified if we compare it to this very extensive database? Well, we do get slightly fewer reads classified within our samples if we just use what is currently known oral diversity compared to the most extensive database we have available, but it's actually not a statistically significant decrease in most of these studies. So basically, increasing our databases non-specifically to just include everything that we possibly can doesn't substantially increase the species that can be identified within our samples. And we need, hopefully, taking a targeted approach can really improve this. So if I instead add all of these novel oral mags to the HOMD database and I profile my samples using the HOMD database, so known plus novel oral diversity, how does this improve the classification rate of these samples? In this case, it actually substantially improves the classification rate. It raises most of these samples to above 50% classification. So we're able to use a much better proportion of our data. We're hopefully able to get a much more accurate representation of the number and kinds of species which are actually present in our samples. In many cases, it approaches 75% classification, which is around what we were getting for our modern samples. So this makes them hopefully much more comparable to our modern samples. And we can do all kinds of diversity analyses and get more accurate results and draw more accurate conclusions from them now. So how does expanding databases with novel taxa alter our ecological diversity estimates? So to look at this, I took my ancient calculus samples, and I profiled them with the HOMD database alone, and then the HOMD database plus these novel mags. And I took the average of each species abundance across all the ancient calculus samples, and I looked at which species were the top 20 most abundant by profiled by HOMD alone versus HOMD plus mags. But before I show that, I need to talk about the taxonomic classification of these species, because we are dealing with now species that are undescribed and they have no names, but we need some way to identify them within our databases so that we can distinguish them in the output. Now, to actually name species, you have to meet a set of criteria that includes growing organisms. So for the most part, with our ancient samples, we'll never be able to formally name these because we can't grow them. So we have to take other approaches. And the one that I settled on was basically to make placeholder names for these species rather than trying to give them names to refer to them, because this is just much quicker and it's much more consistent. So to generate these placeholder names, what I did was to take the genus of each species cluster and use that, and then assign just a primary cluster for PRCL and then the cluster number. So going down the line, just adding the cluster number to each of these. And then this is the naming classification scheme that you're going to see on the next couple of slides. So even in cases where we have species that do have names, I overwrote those with this cluster number just to make everything consistent. So if you are familiar with the oral bacteria, you won't see any species names that you know, you'll just see these primary clusters within the next couple of slides. So here I'm showing the top 20 most abundant taxa averaged across all of these calculus samples when I profile with the HOMD database. So the average abundance is represented from the HOMD database with these light purple circles, but I've also shown as a comparison the average abundance of each of these organisms profiled with the HOMD plus mags database in dark purple triangles. And of course, they're ranked from the most abundant down to the 20th most abundant at the bottom here. And what we see is a profile that's very much like what we have been seeing consistently within our ancient calculus samples to date with the databases that we have. All of these are well-known oral taxa. The first and most abundant organism is Flexlinia primary cluster 3350. This is the Abbit 439 reference genome, demonstrating just how prevalent and abundant this organism is, although there is, of course, a wide variation across calculus samples. But this is essentially exactly what we expect to see based on the data that we have so far. Now, if we look at the top 20 most abundant taxa, if we profile the samples with the HOMD plus MAGS databases, what we see is a little bit different. So there's only 20 species here, which are in the most abundant, or there's only 6 species in this 20 most abundant list, which are overlapping between HOMD and HOMD MAGS. And those are shown with the stars here. But you can also tell because they have a light purple circle and a dark purple triangle. Any of the species that have only a dark purple triangle are novel species that come from our MAGS, and these do not have representation in the oral databases currently. So what we can see here is that except for those 6 species which are found in the HOMD database, everything else in the top 20 most abundant is a novel species. So this is quite a distinct change in what we see as the most abundant taxa, particularly regarding the first most abundant taxon, which is an organism that actually has no genus and species name within an NCBI. So this is another one that's just essentially got a placeholder name. It's an organism that falls within the Firmicutes phylum, but other than that, we know nothing about this organism. But the other taxa that we see, these novel taxa, do belong to well-known, well-characterized genera from the oral cavity. So things like Desulfomicrobium, Methanobrevibacter, Olsonella. We do have current representatives of these genera within our oral databases, but clearly the species here are novel species that have not yet been described. And yet these ones are the most, or the more abundant ones within our calculus samples. So when we profile with this expanded database, we do change the species profiles that we're seeing. And hopefully then moving forward with diversity analyses using these kinds of profiles, we'll be able to see changes between sample groups that we're interested in that we may not have otherwise been able to identify because we don't know the species, whether these species are changing in abundance or presence or prevalence, if we don't know that they're there to look at them to start. So the last part of the talk, I'm just going to talk a little bit about the species which are present only within our ancient samples. So are there species that are unique to ancient Homo? They're not found in modern samples. So yes, there are. And I'm showing that in this upset plot here, which is essentially like a Venn diagram. It's a way of showing a comparison, in this case, of species that are shared between the groups that I'm interested in. But it can be a bit confusing to look at if you're not familiar with it. So I'll walk you through it. So what I'm looking at here is shared species between 4 groups: modern humans, ancient humans, Neanderthals, and non-human primates. And what we have at this end of the plot is showing the species clusters in total that are found in each group. So modern humans have more than 3,000 species, ancient humans have over 300, and there are fewer in Neanderthals and non-human primates. And the total number of species clusters that we see within these groups is probably just related to the total number of samples, which is indicated over on this side of the plot. We had the most samples coming from modern humans, which makes sense. We just have many more samples from living individuals. We had many fewer samples from ancient individuals, Neanderthals, and non-human primates. But then the important part of this information, or this graph, is in the center plot here, where we have per column the number of species that are shared between the groups, which have a dark circle and a line connecting them. So within the first column here, we have 4 species that are shared between modern humans, ancient humans, Neanderthals, and non-human primates. In the second column, we have 9 species shared between modern humans, ancient humans, Neanderthals, but not non-human primates. In the third column, 21 species shared between modern humans, ancient humans, and non-human primates, but not Neanderthals, and on down the line until you get to the single black dots at the end here, which tell us how many species are unique to each of these groups. So modern humans have 3,100 species that are not found in any of these other groups. Ancient humans have 145 species, which are found only in ancient human samples, not in these other groups. So if we look together at the ancient human and Neanderthal samples, those that I've highlighted in orange here, it turns out there are 169 species, which are currently only found within ancient samples. So this is a lot, apparently, of novel diversity, which we currently have no modern representatives of. What species are they? Many of them are unknown. They don't have a species name. Some of them do, which suggests that they're actually misnamed and they need to be reclassified as their own species. Are they all from a limited number of higher level clades? No, they're actually from across the microbial tree of life. So this is quite a wide array of diversity, which currently is only known to exist within our ancient samples. And are these examples then of extinct microbial species? And I would say at this point in time, it's too early for us to say because we don't yet have sufficient sampling of living populations. And as an example of this, from those plaque samples from populations in Cameroon, it turns out there were 31 MAGs assembled from those populations, which are found also in our ancient samples, but in no other living modern human populations that we've sampled to date. So if we had never sampled the individuals from Cameroon, we would assume that those 31 mags are only found within our ancient samples. But with our broader sampling efforts, we know that they still exist in populations today. And I think that as we do more broad global sampling and sequencing efforts, we will see that the number of species which are unique to our ancient samples will decrease over time as we see that there are representatives of these species still existing in populations today. They just haven't yet been sampled and sequenced. So it's really too early for us to discuss whether or not there are possibly species that have gone extinct from our ancient calculus samples. So just to summarize what I've talked about today, diversity estimates depend very much on database completeness. And incomplete databases can lead us to severely underestimate diversity within our samples, particularly within ancient dental calculus. So incomplete databases can cause misleading results and interpretations, and we do need ways to improve our database completeness. And the method that has been taken within the field of metagenomics and microbiome studies has been to generate metagenome assembled genomes directly from samples to avoid reference biases. We know looking at mags from oral samples, both ancient and modern, that there's really a high amount of undescribed, uncharacterized diversity within these samples. And adding these mags to databases should help us reduce our reference biases and improve the taxonomic resolution that we get with our samples, and hopefully allow us to create more accurate diversity estimates and get more appropriate understandings of how oral microbiome diversity has changed through time and across geography. And then just to wrap up, since my talk was very microbiology focused, and I want to touch on how this is actually relevant to archaeology. So why does resolving oral bacterial diversity matter? So oral bacteria are inherited through close contact, and close interactions are difficult to trace archaeologically. So if we can reconstruct specific species and strains directly from our ancient calculus samples, we may be able to use these specific strains to track introduction and loss of these organisms, to look at how changes in social dynamics within and between human groups have occurred, particularly regarding the adoption of social and cultural practices such as caregiving and meal sharing, which are instances where we do know today that oral microbial species are exchanged. Further, it's possible that we may be able to track migration of human populations using the microbes that they were carrying with them in instances where we cannot get tracking of the humans themselves, because calculus actually preserves DNA a bit better than a human bone sample does. And we find really well-preserved calculus DNA in areas where we actually have very poor preservation of human DNA from human bone samples. So we may be able to track the migration of individual species that were carried by humans in situations where we cannot track the migration of the humans themselves. And so with that, I would like to wrap up and thank my group and my funding sources and all of you, and I'm happy to take questions. Thank you.